We're still good on the screen, is that correct? You can still, awesome, fantastic. All right, let me know, basically unmute yourself and tell me if something is going wrong. It's gonna be fine. All right, sterile collection, as we mentioned. You don't want to inoculate, to contaminate your medium, your sample, by any unknown microorganisms that can reside in your non-sterile instrument, God forbid. So, medium. We discussed the concept of medium, anything that supports microbial growth. And you will see many examples as we go through different systems of classification for the medium. So first classification approach is based on the physical state. So liquid medium, liquid, room temperature, can contain a variety of nutritional, let's say, mixtures derived from soy, like trypticase, soy, agar, derived from yeast extract. There are media based on milk. It can contain blood for certain reasons that we will discuss. But basically, the main idea, it can allows you to, first of all, accumulate large amounts of microorganisms. You can easily cultivate liters or hundreds of liters of liquid medium that contains bacteria, for instance, or viruses, and produce an enormous amount of microorganisms. It can serve as the indicator medium. So for instance, in this image in the bottom, you can see uninoculated control on the left. Once you inoculate this liquid medium that contains litmus indicator that changes its color. I don't remember if it's litmus or phenolphthalein. The point is, this medium is red because it contains pH indicator. When you inoculate it with the fermenting bacteria, the result of fermentation, in this case, is generation of lactic acid. Lactic acid decreases pH, and decreased pH is followed by the change of the indicator color. So you see progressively it goes orange and then yellow. So liquid medium is really convenient way to detect bacterial growth based on different indicator approaches. On the other end of the spectrum is the solid medium. You can see an agar, I believe it's a telluride agar, but I'm not sure 100%. You can see this agar plate. So it has a solid surface, which allows you to characterize colonies from the standpoint of their appearance. When you grow bacteria, you can say, on this agar, my bacteria grow as the round, sharp-edged, convex colonies, I don't know, white color, whatever, motile, non-motile, glossy. So you can do a lot of characterization, a lot of descriptive analysis of the appearance of your microorganisms. For the convenience, we use agar, which then, actually it can be found in various, what's the proper word, like baking products, I guess, like jellos. It's basically a derivative, it's derived from algae. It's a powder, you can buy it in a grocery store. If you mix it with water or a buffer and you heat it up, it's called melt, not really melt, well, kind of melt. And then when you pour it into the petri dish like this, it will solidify. This is why we often call the agar medium liquefiable solid medium. You can liquefy it. Another that's not a compound, material. Another material that can be used for bacterial cultivation is gelatin. If you decide to do a home-based microbiology and do gelatin petri dishes, beware, some bacteria can eat gelatin, so in the end you may end up with no medium and no bacteria in your plate. Between the liquid and the solid medium, there is a semi-solid medium without a solid surface. I mentioned yesterday, it is like a clot. So here in this image, you can see 4 so-called slant tubes. They're called slant because, well, the surface is slanted. Now, if you will hypothetically take this tube, turn it upside down, and leave it like that for a few days, the medium will slowly drip out of that tube, very excruciatingly slowly. So really, like, nobody does it. So what are main uses for semi-solid medium? Obviously, it's not good for accumulating large amounts of microbes. How are you going to filter them out? How are you going to separate them from the medium? And actually, neither is agar. Agar is not good for accumulation. It's good for characterization and isolation. Okay, so if you can't accumulate large amounts of microbes in the semi-solid medium, what do you use it then for? First of all, you can determine the motility of microorganisms. So how would you do that? Imagine that this is the tube with semi-solid medium inside. What you would do, you would take a wire, sample your bacteria, and inoculate your medium in the vertical motion in the center. Essentially, you create a trace of bacteria in the very center of your medium. If your bacteria cannot move, you will observe growth exactly where you inoculated. However, if your bacteria can move, then you will see the spread of bacterial growth to the sides. I hope it makes sense. Another useful application of semi-solid medium is to identify different bacterial species based on the growth appearance. And this is one of my favorite examples, really. So these 4 tubes all represent so-called triple sugar iron agar, or iron slant. So what does that mean? Triple sugar part refers to the presence of 3 different kinds of sugars, carbohydrates: glucose, lactose, and, if I remember it correctly, mannose, but that's not so important. The third one is not important. So glucose and lactose first and foremost. Now, iron refers to the presence of soluble iron salts. It's 3-valent iron. I'm not going to ask the valence, believe me. Don't worry about it. So here's the point. On the right, you can see uninoculated medium. That's pretty clear. So what's on the left? On the left, you can see a slant inoculated with the bacteria that can ferment glucose, ferment lactose, and on top of that, run aerobic respiration producing CO2. That is essentially will be E. coli. You know, intestinal bacteria, so on and so forth, right? This one appears slightly different, and you can see the change in color, right? Change in color, it's yellow because of the fermentation. This empty space at the bottom of the tube, that's CO2. So that's very characteristic appearance. Next one, this one. Yellowish, only glucose fermenter, no lactose fermentation. Okay, or very weak lactose fermentation. and no CO2 production. So that is different. That will be either Serratia marcescens or Enterobacter aerogenes, also two microorganisms that can be found in the human GI tract. And finally, this one that appears totally black. So why it is black? It turns out that some microorganisms, namely Proteus mirabilis and Salmonella typhi, I believe the name Salmonella sounds unfortunately very familiar. Both these genera of microorganisms can generate hydrogen sulfide. Hydrogen sulfide binds to 3-valent iron, and that leads to the formation of iron sulfide, which is A, non-soluble, B, black. Basically, there is a video, if you're curious, there is a video in the lab playlist with the lab, I believe it's lab number 10, called EntroTube. I mean, it's not geo or anything, you can just watch it. And in this video, I discuss one of the tubes, EntroTubes, that have this very characteristic hydrogen sulfide black appearance. It's like, you see that, you immediately say it's either Proteus or Salmonella. One out of two. No doubt. Nothing else can do that. Okay? So that's convenient. So next topic of conversation are classification of media by composition and by purpose. So composition. Complex. Sorry. Anyway, it's good. Complex medium. Peptone, yeast extract, milk, beef and heart infusion, for instance. Yes, there is such a thing. So bovine heart, not beef, brain and heart infusion, sorry. So bovine brain and bovine heart are dried out and then can be dissolved in water to produce medium. Obviously, when you take heart and brain and dissolve it in water, that's going to provide a lot of nutrients. So complex media are great to cultivate a variety of microbes from the sources. You don't know what's going to grow there, but something will. Actually, I plan on inoculating some of the agar plates for environmental samples to see, what I can isolate from my own house. However, you can't truly control the nutritional composition. For instance, different batches of yeast extract or different batches of brain heart infusion, they may differ slightly because, well, yeasts are different, cows are different, so that may affect the outcome of your experiment. To avoid this, to control for the variable of the nutritional composition, scientists use synthetic medium. I mentioned it yesterday. You take buffer with a known, well, you take water. You start with water. You add salts, you add carbohydrates, amino acids, various chemical components, which you can exactly measure, the amount of which you can exactly measure. So at every given moment, you know what is in your medium. And then you can add, remove, alter, change the amounts, and therefore you can see how medium composition affects the growth of microorganisms. That's basically the main purpose. Now, based on the purpose, media can be divided into general purpose media, TSA, trypticase, soy agar, a whole slew of media grow, okay. Enriched. This media can support the growth of fastidious microbes. I mentioned Neisseria, meningitis and Neisseria gonorrhea yesterday. plague, Yersinia pestis is an example of a fastidious microbe. Um, they may require various growth factors or essential nutrients. So, for instance, for Neisseria, blood or chocolate agar. Both of them contain blood in either denatured or non-denatured form, meaning that they contain a lot of iron. Now, selective medium um, blocks growth of certain microorganisms. For instance, mannitol salt agar contains a lot of salt. Salt is not really conducive to bacterial growth, except if bacteria are what's called halophilic. And interestingly enough, Staphylococci, bacteria that we discussed yesterday, are indeed halophilic. So you can use that mannitol salt agar to selectively culture Staphylococci while inhibiting the growth of other microorganisms. Another good example is EMB agar, which contains two dyes, eosin and methylene blue, and these dyes inhibit the growth of gram-positive bacteria. Now, unlike selective medium that selects for a certain kind of microorganisms, differential media allows you to distinguish between different microbial species, distinguished by their appearance. So, for instance, on the blood agar, which contains intact, basically, sheep red blood cells, you can see the difference between various microorganisms in terms of their ability to destroy red blood cells. Some of them will completely obliterate any traces of red blood cells, and we call such microorganisms beta hemolytic. Some of them can partially destroy red blood cells, and they will be alpha hemolytic. And some bacteria will not do anything. I mean, they will grow, red blood cells will still remain intact. We call them gamma hemolytic. McConkie agar, for instance, allows you to distinguish bacteria that can or cannot brain fart, I'm sorry, can or cannot ferment lactose. So many of them grow, some of them ferment, some of them don't. Now, the question sort of that you may have now about selective and differential media. Can they be both? The answer is sure. For instance, McConkie agar very strongly inhibits gram-positive bacteria, allowing the growth of gram-negatives. Is it selective? Yes. It also allows you to differentiate between lactose fermenters and non-fermenters. Is it differential? Absolutely. It's both selective and differential. So the bacterial cultures that you obtain in your medium, whether it's liquid or solid or semi-solid, can be pure or xenic, single species. We will try to produce an xenic culture during a small part of the lab number 4, which is the strict plate. They can be mixed. That's what I expect to see on our hand wash sample. I'm kind of hesitant to check on it. I really want to see growth, but I don't know, hopefully. It's not incubator, so I'm kind of and they can be contaminated, undesirable microbes. Unfortunately, it's a damnation of the microbiological lab to avoid contamination. It's really hard. Now, another question that we need to answer is the cultivation of viruses. How the heck do we do that? Viruses, as I mentioned, are obligate intracellular parasites. So for a very long time, they were cultivated in animals. I mean, animals consist of cells, right? You can inoculate animals and viruses will grow in the cells, but that's a pretty darn expensive method to grow viruses because animals are expensive. So in the late 40s, early 50s, Italian scientist who worked in England, Renato Dulbecco, developed a revolutionary method that is called a cell culture. So what it is exactly? On the right on the slide, you can see a set of flasks that sit on the side, and inside of the flask, you can see a red liquid. So I can tell you right away that on the well, kind of on the bottom of this flask, each flask here, on these sides of the flasks, there will be cells growing in a single layer. So they grow on the plastic. And this red liquid is nutritional medium that supports cellular growth. But the point is, you can add virus to these cells. Wait, no, oh, can you see that now? Yeah, sorry, it was at the very bottom. Sorry. Yeah, so this is the virus thing, okay? So, um, at the you can add viruses, okay, and viruses will infect those cells, and the cells will grow. That's basically it, okay? Not cells, sorry. And viruses will will grow in those cells, and you can collect them because viruses will leave the cell, and you know. So, but the point is, you really need cells to replicate viruses. This is why developing a cell culture model is so important and so crucial for virus cultivation. You can just take virus, throw it in the medium, and then say, "Huh, yeah, that'll work." Actually, one of the interesting questions that we discuss when you hypothetically when you tap the TSA agar to see what's on your hands, you can detect bacteria, you can detect fungi. Fungi will grow in TSA as well. But viruses, you cannot, because there are no cells that viruses can grow in. Does that make sense to you? Okay, before we kind of cut the recording off, any questions? Okay.